In this video, we will focus on explaining cyclic and non-cyclic photophosphorylation. Sun is the major source of energy on Earth. Light energy enters food chain by capturing photons by primary producers that contain chlorophyll. Chlorophyll converts light energy into chemical energy through a process called photosynthesis. So what is exactly photosynthesis? It is a process of synthesis of carbohydrates from carbon dioxide and water using light energy. Photosynthesis is a two-stages process: light-dependent reaction and light-independent reaction. Before we start talking about the process, we need to understand some terms and minor processes to make it easier to understand the whole image. Antenna complexes. It collects radiant energy or light to excite center reaction. It composes of chlorophyll, accessory pigments, and proteins. There is one antenna for each photosystem. Photosystem 1. It is a complex of molecules to excite electrons, preparing them for the electron transport chain. It composes of center reaction chlorophyll, called P700, antenna complex, and other pigments such as ferredoxin, phylloquinone, and some proteins and ions. Photosystem 2. It is almost as similar as photosystem 1. It composes of center reaction chlorophyll called P680, antenna complex, and other pigments such as phosphatin, plastoquinone, and some proteins and ions. The main differences between photosystem 1 and photosystem 2 are that photosystem 1 contains P700 and exists in both eukaryotes and prokaryotes. On the other hand, photosystem 2 contains P680 and exist only in eukaryotes. There are other complexes like cytochrome B6F complex. It has an important role in electron transport chain as it contains iron-sulfur protein as well as other proteins and ions. Oxygen evolving complex. It is a part of photosystem 2. It helps in photolysis of water into free electrons, free protons, and oxygen molecule. So, why does the process need that? Simply, it needs electrons for electron transport chain and needs protons for chemiosmosis. Chemiosmosis is the movement of protons with the gradient of concentrations of protons across the thylakoid membrane. As the outer stroma is basic as pH 8, and the inner lumen is acidic as pH 4, due to the activity of photosynthesis, ATP synthase or ATPase enzyme, it generates ATP molecule using ADP and phosphate group in a process called photophosphorylation. Photophosphorylation shortly is the use of light energy to convert ADP into ATP. It takes place by one of two methods: cyclic or non-cyclic. Starting with non-cyclic, it takes place in grana, specifically at thylakoid membrane. A photon of light enters photosystem 2 and splits water into hydrogen ions and oxygen by photolysis, with the help of OEC. The accessory pigments feed the light energy into the reaction center, where two electrons are excited and leave the reaction center and are accepted by a primary electron acceptor. The electrons are then passed down an electron transport chain. While the electrons are passing through the cytochrome, protons enter the stroma, allowing the ATP synthase to pump protons out again. But this time, it catalyzes the production of ATP from ADP, as we explained in chemiosmosis. The excited electrons then enter photosystem 1, where the energy from light excites electrons and leave the reaction center and are accepted by a primary electron acceptor. The electrons pass down another electron transport chain and are used to reduce NADP into NADPH. And that is how non-cyclic photophosphorylation takes place. The final products from non-cyclic is ATP molecule, NADPH molecule, oxygen molecule. Cyclic photophosphorylation. Although it is similar to non-cyclic, it is much simpler, as it takes place in photosystem 1 only. A photon falls on photosystem 1, releasing two electrons by the water photolysis. The accessory pigments feed the light energy into the reaction center, where two electrons are excited and leave the reaction center and are accepted by an electron acceptor. The electron passes the electron transport chain as the following. The electron is being passed from the primary acceptor to plastoquinone, then to cytochrome, then to plastocyanin, before returning to the chlorophyll again. This transport chain produces a proton motive force, pumping hydrogen ions across the membrane. This produces a concentration gradient that can be used to power ATP synthase during chemiosmosis. The main differences between the two processes are the photosystems that participate in, and the end products, as the cyclic produces only ATP without NADPH. That's all for today. Hope we helped you to understand this topic. Thanks for watching.